Hi everyone, it's Dr. LeBlanc with Unit 8C, Recombination and Transposition, Transposable Elements. Our learning goals are to understand what transposable elements are, to understand the structure of transposable elements, to understand the mechanism of transposition, to understand how transposable elements are organized and distributed in a genome, to understand the consequences of transposition on gene regulation, to understand how transposition can cause mutation, and to understand how transposable elements may have influenced evolution. This is a picture of variegated corn kernels on an ear of corn. The speckling phenotype is due to the movement of DNA segments called transposable elements. There are many different types of transposable elements that have been identified in everything from bacteria to humans. This is a partial list, and only a few examples of bacterial transposons, how long they are. Some of them are pretty long. Some of them are very short. From yeast, the Ty elements, from Drosophila, the P elements and copia-like elements. And humans have two basic types, short and long. Within the genome of many different organisms, transposable elements can comprise a very large percentage of the genome. For example, in Xenopus laevis, a frog, there are 77% of the entire genome is comprised of just transposable elements. Humans themselves, 45% of our genome are transposable elements. There are two different pathways for transposition. That is, the moving of the DNA element from one location to another. First is simple transposition, and a corollary of that, simple transposition coupled to DNA replication. And then there's retrotransposition, which occurs via an RNA intermediate. Simple transposition is just like moving a mobile home. You settle down for a little while, and then you move, you pull up your roots, and you move to a different location within the genome. Transposase is the enzyme that mediates the moving of a transposable element. So here we have a transposable element. It has DNA in the middle, and it has DNA on the ends. The DNA on the ends is characterized by inverted repeats. That would mean you have ATGC here, and again ATGC here in the same orientation. Transposase is the enzyme that mediates the removal of the transposable element from one location and the integration of it into the genome in another location. So transposase catalyzes the removal and its reinsertion. Transposase recognizes the inverted repeats at the ends of the TE and brings them close together, loops out the DNA. Here it is. Okay? So it brings the two ends together, and then it cuts the DNA. Transposase cleaves outside of the inverted repeats, right here, and there's a little bit of DNA that's removed. This excised transposable element then can insert itself into a new chromosome location. When it does that, it leaves little telltale sequences at the ends. Okay, so those are called direct repeats. Here is an example of transposition that's a little more up close. So initially, transposase cuts the DNA at the target site, a random spot in the genome. Here the sequence is ATGCT, cuts on either side of this certain number of base pairs. Then the transposable element is inserted into the target site, and then you have DNA synthesis to fill in this gap here. So when a transposable element integrates into a new site in the genome, it has a telltale signature of direct repeats. ATGCT, ATGCT on either side of the integration site. They're called direct repeats because they're the same, they're repeated in the same direction. Okay, here's a couple of different examples of transposable elements that move by simple transposition. So here is a transposase gene that is encoded by the transposable element. So the actual transposable element is right here. Okay, it includes the inverted repeats. The direct repeats are the consequence of integration at this site. Okay, and again, here is a transposable element. This one's a little bit longer. This is an example of one that we can find in bacteria. It harbors a transposase gene, so within this sequence you can see the relic of the original transposable element. Here it is right here. So this one looks just like that. But along the way, it's also picked up another transposable element, a piece of DNA that has an antibiotic resistance gene. There are many transposable elements with resistance genes in them, and that is not good news for patients in the hospital. So simple transition coupled to DNA replication can increase the number of transposons per genome. How is that possible? Well, here we have a replication fork. Okay, you're quite familiar with that. And it has just replicated a transposable element. So there was a transposable element, and we've replicated it so that both new strands of DNA, the two daughter strands, have a copy of the transposable element in them. If a transposable element moves from here to here, okay, this strand of DNA that goes from here all the way down after it's been replicated will now have the TE here, just like this, okay, but it will be missing from over here. However, this other strand of DNA, this one, had a transposable element when it was originally replicated, but now it will also have a second copy of the transposable element after the replication fork makes its way up the chromosome some more and encounters this newly transposed element. So this DNA, this daughter strand of DNA, will have two copies of the transposable element. So DNA replication has proceeded past the point where the TE has been inserted, and the top copy of the TE then transposes ahead of the fork where it's copied again. Okay, what is retrotransposition? Retrotransposition occurs via an RNA intermediate. So you have a transposable element in the genome, and you transcribe it into RNA. Okay, this RNA right here, transcription into RNA. Then another enzyme called reverse transcriptase, which has its origins in viral genomes, makes a DNA copy of the RNA. Okay. It's first transcribed into DNA, and then the RNA is degraded, and you copy the DNA into double-stranded DNA. Then a second enzyme called integrase nicks the DNA and can nicks the DNA of the chromosome and inserts this new little piece of DNA that originally came from a transposable element. So in the end, you have multiple copies of the transposable element within a genome. Where do these enzymes come from? Reverse transcriptase and integrase? Well, they're actually encoded in the transposable element itself. These elements are often termed retroelements, retrotransposon, or retroposons. I usually go for this middle one, retrotransposons. So here's a diagram showing what a retrotransposon looks like. Okay. At the ends, it has long terminal repeats. Okay. And they are direct repeats. Also at the site of integration, we also see direct repeats, but those are not part of the TE. They are a relic of the integration. Okay, so we needed a reverse transcriptase and we needed an integrase. Both of those are encoded within the transposable element itself. This is an LTR retrotransposon. There's also some non-LTR retrotransposons that have a different combination of those genes. So transposable elements can move by retrotransitions via an RNA intermediate, and they carry within them the genes that they need. Here are some examples of transposable elements again. So the two that we're going to talk about for humans are both retrotransposons. Okay. In bacteria, we have mostly transposons. In yeast, it's a retrotransposon. In Drosophila, we have one of each. And plants also, there are examples of one of each. Transposable elements have influence on mutation and evolution. They probably occur in the genomes of all species. So far, we haven't been able to find genomes that don't have them. They can rapidly enter genomes and proliferate. There are many different kinds, and they can be a major part of the genome, as we had already seen. Mammalian transposable elements, in humans, they're called lines and signs. Lines are long interspersed elements. Okay, so they're longer elements in this category. They're usually 1,000 to 10,000 base pairs long, up to 10,000 base pairs long for one single line element. And they're found in a few thousand to several 100,000 copies per genome. In humans, it's on a higher amount. Signs are short interspersed elements that are less than 500 base pairs in length. For an example is the ALU sequence. The ALU sequence came about from reverse transcription of a small cytoplasmic RNA that has a very important job called the 7SL RNA. The 7SL RNA in the cytoplasm helps proteins that are going to be synthesized through the endoplasmic reticulum and processed through the Golgi and eventually either embedded in the membrane or deposited outside the cell. The 7SL RNA is part of a particle called the signal recognition particle that anchors ribosomes to the endoplasmic reticulum and helps in the process of translocating the newly growing polypeptide across the membrane of the endoplasmic reticulum. So it's a common small RNA found in the cytoplasm. Somehow a reverse transcriptase got a hold of it and turned it into this ALU short interspersed nuclear element. It's present in approximately 1 million copies in the human genome. So here's an example of an organization of some of the transposable elements within a gene. The green, dark green here, are exons. The light green areas are introns. And the purple and blue and yellow elements here are ALU elements, other signs and lines. So you can see within this one long gene, we have many, many, many transposable elements. And if you look carefully, they are all present in only the intron sequences, not within the exons. Transposons can have a variety of effects on chromosome structure and gene expression. They can insert themselves into genes. They can delete genes when they remove themselves. They can also cause inversions. We can indirectly see the effects. They can result in chromosome breakage, the chromosome rearrangements, changes in gene expression, changes in gene regulation, gene duplication, and changes in exon content if they happen to pop themselves into an exon. The biological significance of transposon in evolution remains a matter of debate. This is another list of what I just went over. So, for example, one of the possible consequences of transposition are changes in gene expression. You can cause a mutation by incorrect excision of a transposable element. You can have gene inactivation if a transposable element inserts itself into a gene. This has been witnessed a lot in Drosophila. There are many, many documented examples of this occurring in Drosophila. You can see alterations in regulation when the transposition of a gene next to a regulatory sequence or the transposition of regular sequences next to a gene. So moving elements around the genome can cause significant consequences for gene regulation. And of course, you can mess up a gene by inserting yourself into the middle, and you can even duplicate genes if they're attached to transposable elements, and you reverse transcribe them and then move them to a new location. Here's a very interesting report from the journal Cell. The question of how organs arise, new organs, during the course of evolution has always been a little bit of a quandary for developmental biologists and molecular biologists. This report may shed some light on how a brand new organ can evolve from species that don't have it. And the title of this report is Ancient Transposable Elements Transformed the Uterine Regulatory Landscape and Transcriptome during the Evolution of Mammalian Pregnancy. So this is a summary. I'm going to just read it very quickly. How morphological novelties originate. Morphological novelties are things like new organs, like a uterus, is a major unanswered question in developmental and evolutionary biology. These folks demonstrate that thousands of genes evolved uterine expression during the evolution of mammalian pregnancy, and that the recruitment of the new gene expression in the uterus was likely mediated by ancient mammalian transposable elements. So it seems like if you need to make a uterus, you need to have a whole lot of genes expressed in the uterus, and that may have been mediated by transposable elements jumping down into a variety of genes and coordinating their expression all together because it was the same transposable element. And that's the subtitle of this figure, Ancient Mammalian Transposable Elements Coordinate Uterine Expression During Pregnancy. You can look more of this up at this website. So in summary, transposable elements are small mobile genetic elements within the genomes of both prokaryotes and eukaryotes. TEs are dispersed throughout the genome of many organisms. TEs comprise a large percentage of the genome of many species. During simple transposition, a TE moves from one site to another in the genome. Transposase cuts out the transposable elements and cuts the target sites, leaving single-stranded ends. Direct repeats are created at the site of integration. Bacterial TEs can carry antibiotic resistance genes. When transposition occurs during replication, it can result in duplication on one of the newly synthesized DNA molecules. Retrotransposition occurs via an RNA intermediate. Reverse transcriptase makes a copy of the TE and cuts the chromosomal DNA and inserts the new TA into a new site. Some retroposons have long terminal repeats at their end and encode their own enzymes for transposition. The human genome has two major types of TEs: lines, long interspersed nuclear elements, and signs, short interspersed nuclear elements. Transposition can interrupt gene function and genome integrity in a variety of ways. Transposed elements have influenced evolution, such as the evolution of the mammalian uterus.